Good morning, everyone. We are from Group 1 that consists of 4 members, namely Alnail Marisa, Desita Yolanda, Christian Felix Silalahi, and Rio Madani Sitompu. So here, we would like to present our critical journal review with the title Origin and Evolution of Endoderm and Mesoderm. So first, I will explain about the identity of journal. The title is Origin and Evolution of Endoderm and Mesoderm. The journal is the International Journal of Developmental Biology, number 47, pages 531 until 539. Year in 2003. The authors are Ulrich Denau and Corinna Biscols. Date of review on 12 April 2020. Next, we move to the introduction. More than 30 of the 35 described animal phyla develop from 3 germ layers, the outer ectoderm and the 2 inner layers, endoderm and mesoderm. Beside these triploblastic animals, few phyla exist that consist of 2 germ layers, endoderm and mesoderm only. The evolutionary origin of endoderm and mesoderm is closely linked to the origin of metazoa, i.e., the transition from protist to metazoa. Since multicellularity is accompanied with the division of labor of the cells, those that are specialized in digestion become located in the inner side of the organism, and those that are specialized in protection, locomotion, and sensing the environment remain on the outer side of the organism. Further, since the next major transition from diploblastic and radially symmetrical to the triploblastic bilaterally symmetrical animals also marks the origin of the second body axis. The emergence of the mesoderm is also linked to the evolution of axis formation in metazoa. The 2 inner layers, endoderm and mesoderm, arise during or tightly linked to the gas relation. Next, we move to the problem solving of this journal. In order to understand the evolution of endoderm and mesoderm, the authors have to understand gas relation and its molecular regulation. In the first part of this review, they will therefore summarize different modes of gas relation that have been evolved in the different phyla. In the second part, they will examine the role of some crucial conserved genes involved in the formation of endoderm and mesoderm in bilateria, and discuss whether the evolutionary origin can be traced back by analyzing these genes in basal metazoa. Next is research purposes. The evolutionary origin of the inner germ layers, endoderm and mesoderm, and their relationship have been a matter of debate for decades. So, in this review, the authors summarize the major modes of endoderm and mesoderm formation found in metazoa, and possible evolutionary scenarios to reconstruct the ancestral state. In the second part, the authors address the question whether endoderm as well as mesoderm are homologous among bilateria. Next is research subjects. This research focuses on 4 classes of genes: the ZF finger gene GATA4-6, the BHLH gene twist, the crupple-like ZF finger gene SNIL, the T-box gene brachyury. Next is research methods. The authors propose that the comparative analysis of some crucial transcription factors involved in the early specification and differentiation of these germ layers might provide cues for the level of homology. data was taken from literature about the study of Endoderm and Mesoderm.
Okay, so now I'm gonna explain about the data analysis. So this research data is analyzed by using descriptive method, which means all of the literature this research find about the Mesoderm and Endoderm are compared, and then from that comparison we will find the result. The result will be told descriptively. So that's why we can say if this data analysis use descriptive method. Other than that, also this data analysis is using qualitative method because there is no statistic analyze we use in this study, so it's just the comparison between one literature to other literature, like that. So for the next slide, it's about research result. From the data that we analyze, we can find if the result there are 5 points. So the first point is the modes of gastrulation are different between phyla, and then the second is mode of gastrulation in hydrozoa is mesenchymal mode, and the third is endozoan form endodermal mesenteries by epithelial folding, and then the fourth is muscle cells, the major derivative of the mesoderm, differentiate on one side of the mesenteries, and the fifth, there is genes involved in gastrulation and of their molecular network. Okay, so that's what I can explain.
Now we move the part of the discussion. The discussion with what are germ layer as definition and some cautionary note. The first slide. What are germ layer as definition and some cautionary notes? Germ layer are distinct cell layer that form very early during embryonic development and that give rise to all tissue of the adult. For the instance, ectoderm will give the rise to the nervous system and the epidermis. Is it the same word epidermis and endodermis? It's up to letter. The endoderm to the midgut and some mesoderm to the blood. Muscle tissue and vertebrate and the bones and some inner organ is seen. And even in higher animals, ectoderm and endoderm derivatives are often also constantly proliferating. However, differentiation is usually restricted to a particular fate. Next, gastrulation, the embryological stage of the germ layer formation. We talk about the modes of the gastrulation. Haeckel and the others research as the ancestral model and gastrulation founded the epiboly or evangination. Is found in yolk-rich embryos and involved to the migration and proliferation of the cell of the animal pole. So the proliferation and cell of animal polis over the yolk-rich vegetal cells, and this most was proposed as the ancestral mode of gastrulation by Lancaster, who developed his planula theory from this idea. Evangination is always found in well-studied in sea urchin. That review by Intentions 199, Wessel and Wikramayek founded involves of the migration of proliferation of the cell animal pore. Example, epiboly the amphibian Xenopus and the zebrafish. In the events, they said there is a delamination in this gastrulation mode. The inner layer is formed by cell division and the epithelial layer is blastula radial plane. And second is immigration. Immigration is individual cells that can occur from outside from blastula multipolar immigration, or preferentially from the one side and the array embryo oral immigration. And next we talk about B. Gastrulation, the embryological stage of the germ layer formation. In the most animals, endoderms formation goes hand in hand with the formation of the third germ layer, the mesoderm. However, it's not a condition sine qua non of the gastrulation, and indeed mesoderm formation can be temporal. What about the hand-in-hand deformation? So the meaning of the hand-in-hand is the each slide of germ layer, like the first slide and the second germ layer and the third layer, have the formation in there. That's not about conditio sine qua non, but the mean is that who has the explainable power over the seems enjoy unswag look, so like a dogma, and that force mystically influence in the modern persons, so it can be proved. And then we talk about the modes of the mesoderm formation. Example of this found schizozoally is found in several spiralia, where most of the mesoderm development from this descendant of the fourth cell. The cell enter the inner side of the embryo from the posterior margin and the blastopere and proliferates into sac on both sides of the developing gut. Epithelial mesoderms develop directly from the endodermal pouches, which is building the specific places of the ancestron phenolae is spread from the endoderm. So the picture is so weird. Schizozoally, as found many spiralia, specific cell from the margin blastopere as immigrated as single cell. Proliferate and start making coelomic cavity in segmental parts by organism epithelial and forming cleft band twin in the inner cells. Enterocolically, as found the hemichordates in B, parts of the enteron fold the epithelial pouches from the protocol. Mesocol and metacol colors indicate different germ layer. Ectoderm, result in the dark pink. Mesoderm, so in the red color. Endoderm, so in the orange color, and ECM, so in the rose color. Ancestron and coelom in white, and modified by Salvini, Palwen, and Sperata in 1979. Next, we talk about G. The first E among the genes that are commonly accepted as mesoderm markers and key players in mesoderm specification and patterning are those coding for the transcription factor, twist, snail, GAT factors, and brachiorrhea. The role of brachiorrhea in mesoderm formation is well established in vertebrate. And then the if. The least expression of twist in ventral blastoderm shortly before gastrulation. So it's like a twist expression cell make of the ventral furrow, through which is prospective mesodermal cell emigrate into the embryo. That's a twist. And G. Brachiorrhea plays a crucial role in the convergent extended movement and during gastrulation that result the elongation of the body axis in vertebrate. And the E, we talked of the vertebrate and this in invertebrate. Mercury therefore seems to have a dual role in mesoderm specification and axis elongation. In basal, deuterostomes and protostomes, Mercury is expressed around the blattosperm. And we talk about the brachiorrhea part 2, the more explanation. H. The different function of vertebrate and insect. Twist is most likely due to the heterodimer formation with the E proteins. Thereby is competing for the binding E proteins to the McCoBH protein by meioid. In this respect, it is interesting that the twist homologue from the mollusc Patella pulgata, a lopotorozoan pole of the embryo. That's the definition cell that are descended from posterior axial and mesoderm express brachiorrhea. Brachiorrhea part 2. Brachiorrhea is expressed around the blattosperm. And before we call brachiorrhea has played a crucial role that convergent extension movement. And this function is suggests that one ancestral function of brachiorrhea to regulate genes involving morphogenetic movement during gastrulation. In particular, of the cell is preserved mesoderm invertebrate. Their site a peak of brackery. Possible scenario of the fluorination region of Mesoderm by enterozoally. The common ancestors we can find it. You can look in Cnidaria and Deuterostromia is posited by the diboplastic and diploblastic endozoa mesenteris from the folding endodermal epithelium towards the central A. Next, the last of our discussion, that's the neural crest ectodermal cells that mesodermal behaviors. Sikapnya. The cell of the neural crest detach from the epithelium and start the migration as single cell, the mesenchyme of endoderms, mesoderms. That's it, hand in hand. The transition from the epithelial to mesenchymal state is reminiscence of that migratory cell of the mesoderm in amphibians. And verse 2. In both a process is mesoderm formation and neuralization. In both process mesoderm formation and neuralization. The cell that will produce go through AMG expressnil, thus it seems confusing, and the neural crest regulate cell motility and the cell adhesion, similar to the estrol, mesoderm, and formation.
Now we move to the conclusions. So this conclusions is basically the integration between the conclusions presented by the journal and also my perspective, how I view this journal. So taken from the journal, the conclusions is that although the advent of molecular development biology has shed some light in the molecular regulations of endoderm and mesoderm formation, its evolutionary origins remains an open question. This means that although that this research journal was way ahead of its time and can be considered as revolutionary, but at the end of the day, there are some sort of certain limitations, maybe in terms of technology and references in that time. Particularly specifically in 2003. Now, that's why they still say that it remains an open question, because maybe in the future there some sort of development in how we can view things, especially when it talks about the mesoderm and endoderm. So that's why they say that it still remains an open question. A mesenchymal mode of granulation is found in the polar immigrations of endodermal cells in some of hydrozoa, the derived class of Cnidaria. Since the anthozoans, the basal group among the diploblastic, notably muscle cells, the major derivative of the mesoderm differentiate on one side of the mesenteries, which indicates an internal bilateral symmetry of these animals. However, this view requires further supporting data by the comparative analysis of the genes involved in the gastrulations and of their molecular networks. Like I said, that this journal possibly ahead of this time because they giving such advanced information regarding this issue, regarding the origins and evolutions of endoderm and mesoderm issues. But yet again, they still open to the possibility that in the future there might be some development and there can be new findings. So the journal answer in required questions to the findings, and its biggest contribution is to explain the origin and evolutions of endoderm and mesoderm, obviously. The next is the references. Now, the references that this journal used, there are so many of them, but to answer the questions that given from our lecture on how we should talk about the references. So basically, the references used in this journal cannot necessarily considered as up to date, since many of the shown references was from the 90s journals, and the most recent one is no older than 2003. This, however, can be justified due to the date of the research is on 2003, meaning that if we compare this journal to, current findings like the one that around right now, it may be considered as, old school. But yet again, this journal was published in 2003, and of course they can only work from the 5 years top of the journal or references or maybe book that already been made by that era. So the references can be considered as, ahead of its time, but still there is a possibility that things might be changed, and also other things that this can be also become a basis or fundamental things for current research in nowadays. Now we talk about the advantages that these journals have. The journal was ahead of its time and proposed such evolutionary findings. Consider the year of its publishing in 2003. The language is easily read, and the data is not overcompiled with the overall content of the journal like common research journal does. Like there is sometimes a journal, but there are too much like data or maybe tables and graphs without proper explanations, and makes us really confused about what it has to do with the topic of the journal. Since that this journal explaining on how that evolution are in Endoderm and Mesoderm and the origins of it, so it's more like a passage or text-based journal. So I think that this is easily read, considering. Now, this journal giving such a huge benefits to the development of animal development subject, or maybe similar study like embryology, so on and so forth, providing information for future research and developing the related science as a whole. Talking about the benefits to the Indonesian development, of course, at its very least, helping the illiteracy among the society and its best among the scholars. Of course, this research journal, it can be used as a references and also best for upcoming research regarding the issues of the origins of Endoderm or Mesoderm whatsoever. Now, suggestions. As a college student, I don't think that I'm appropriate to giving some sorts of suggestions in terms of the content, but my suggestion is that we can provide it to read future related readings, since this one can only be considered as basis, meaning that like I said, this journal was made or created in 2003. Therefore, there might be right now some sort of future findings and development in regarding this issue. So you can only use this as a references that in that time this was a finding, but it develops in let's just say that 2010, a decade later, things like that. So when you read this journal, you have to read also other journals or other books or other references that close to the time being of how you created your research whatsoever. So that's all about the presentations of our group by Aldehel Marisa, Desita Yolanda, Christian Felix, and Rio Mariani Sutompo. Thank you.